Annual Safeway Pharmacy MS Connections Conference. This is an annual event that happens in this province. It is a provincial conference. Calgary and Edmonton are the two cities that share the honor of being able to host this event. We do it every other year, of course. I am Laura Santos. I am your MC for tonight. I've been involved with the MS Society for about a dozen years, first as a client and as a member, as a volunteer, and just recently as a board member. A couple of housekeeping issues I need to clear up. Washrooms, if you haven't found them, out the doors and back and around, or out this door and around to your left. If you go out this door, you won't be, well, we'll leave the door open so you can get back in here. This is a non-smoking room, but I think most places in Calgary are non-smoking. We have a healthcare aide, Dorothy. Dorothy, could you just stand at the back there? If anyone needs assistance, that's lady's Dorothy, and she'll be able to give you a hand if you need anything from the washroom. Dorothy works for the company Classic Life Care, and they are one of our sponsors and have provided care attendants for this weekend. These sessions are all sold out. The whole weekend's been sold out, and this session especially has been sold out. And it's a good indication that this topic is of great importance for many of us. Carrie Mogden is a member of the Society Board, is a Society Board Chair for the Alberta Division, and Carrie has an introduction for Dr. Stys. So please welcome Carrie Mogden.
Welcome everybody. It's great to see a big crowd out here today. It's a real thrill that everyone has been able to make the time and fight the traffic today. I don't know if everyone else had the same wonderful experience that I did. So, as mentioned, I've been involved with the MS Society since 2003 when I was diagnosed with the relapsing remitting form of MS, and have held a number of positions. I was a member of the Calgary chapter board. Became chair of the Calgary chapter board. I'm chair of the Alberta Division board and a member of the national board of the MS Society of Canada. And most recently was working on the renewal task force that was tasked with this very important work that's just rolling out right now. So I'm thrilled to have an opportunity to be here. I saw Dr. Stys speak at the Alberta Division presentation earlier this year, and I've had the good fortune to travel across Canada and see these presentations, and his was pretty much the best one that I saw last year, so I'm positive that you're going to enjoy it. So I'd like you to welcome tonight's speaker, Dr. Peter Stys. Dr. Stys is a professor in the Department of Clinical Neurosciences for the Faculty of Medicine at the University of Calgary. There's a couple of scientific words in this speech that I'm going to mangle very well because I'm an engineer and I apologize. He's a world leader in detailed study of pathophysiological mechanisms of white matter injury. Was that pretty good? Well, there we go. In stroke and trauma, and recently he's been focusing on the disease mechanisms in multiple sclerosis. Last fall, the MS Society and the MS Scientific Research Foundation announced that Dr. Stys and his team, located here in Calgary at the Hotchkiss Brain Institute, would be recipients of a $3.8 million grant to investigate the complex interplay between degeneration and inflammation in multiple sclerosis. This research is very important because it offers a lot of hope to many people who live with progressive forms of MS, many of whom are in this room today. And who currently there really is no treatment. So I'd like you to welcome Dr. Stys, and I know you'll look forward to his presentation.
Perfect. The AV is working, everything is working. Traffic was heavy as we heard, but we're all here, so that's good. And thank you very much for that very kind introduction. It's a great pleasure to be here and to speak to you this evening. As you heard, I don't come from the field of MS originally. I was a researcher in white matter, and I'll tell you all about what white matter is. So I worked on spinal cord injury, stroke, and that kind of thing. And as time went on, I got more and more interested in MS. Not in the immune inflammatory aspects of MS, because I'm not an immunologist. I don't understand that field at all, and I never will. But I get interested in MS from a different perspective that is becoming more and more popular as time goes on, because while we've made tons of progress with immunology, we've got good drugs for immune suppression, really MS has been stubbornly resistant, at least in the later phases, to our efforts to try to control the disease. So I got interested in it, and I came to this field with a slightly different view, and I'll show you in my talk what that different view is and why we should look at this disease in a slightly different way to try to unravel its mysteries. So I was slated to speak from 7 to 8:30, an hour and a half, but okay. How's that? Is that a little bit clearer in the back? Okay. So I was scheduled to speak from 7 to 8:30, but I assure you, you'll be relieved to hear that I'm not going to go over an hour because I don't know of anybody who can listen to my drivel for over an hour. So it's going to be an hour at most, and I hope you're going to enjoy it. So I'm going to speak to you in 4 points shown here. So we're going to start by reviewing what our understanding, and this is what I learned in medical school, what traditional MS was. I'm going to tell you about white matter that I alluded to, what axons are in myelin, all that kind of thing. You probably know most of that, but we'll go over that. And then I'm going to introduce what I call the fly in the ointment, the theories or our understanding of MS as it stands, what I consider to be important flaws in our perception of what the causes of MS might be based on emerging data. I'm going to just touch upon some of the research tools that we use in my lab and other labs to try to unravel these mysteries. We've been working in this field for 20 years, so of course I can't give you a full overview of what we do, but just a little bit of a potpourri of the kind of techniques and things and approaches that we use to try to get down to the nuts and bolts. And at the end, I'll give you my personal view that is completely unproven, but hopefully you'll share with me the intrigue of why my view of what the real MS, as I call it, might be, may have some validity, and why we should be looking at this disease in a different way, and why this grant that we got will now allow us to do that in much more in-depth and more quickly. So let's start off with the textbook definition. You've all seen this, you've read a lot about it. So we all learn in the textbooks that MS is an inflammatory autoimmune disease of the brain, particularly the white matter, where the nerve axons are ensheathed in myelin, which is the insulation. That gets stripped off by immune cells, and as a result, the nerves can't transmit impulses, and we get deficits. This disease was, in fact, described over a century ago by Charcot, a French neurologist, who really did a wonderful job at describing the clinical features of multiple sclerosis. And we've been trying to unravel its mysteries ever since. We've made a lot of progress, but we still don't know the cause after over 100 years. So we talked about the myelin, we talked about the immune cells, you all know about that interaction. But after all this time, the cause remains unknown, as you see in the bottom paragraph here. And so we have some drugs for MS, but we don't know the cause, and we can control some aspects of it, but we can't cure it. And our approach is to try to understand it in fundamental detail to try to get the root cause. Hopefully by the end of my career or sooner, we're going to get there. So here's white matter. I'm just going to pause here and give myself a pointer because I want to point out to you features here. Okay, I'm going to try to lean into it and speak. Okay. All right, so hopefully this is even louder down there. So I'm going to lean into the mic because it doesn't want to cooperate with my 6-foot frame. Anyway, so here's what white matter looks like. So white matter is, or at least has been, the focus of MS. So here's a MRI of a human brain cut coronally, as we call it, so as if you took a knife and sliced it and looked at somebody from his or her face. So underneath, inside the brain, this lighter stuff is the white matter. That's where all the axons and myelin lie. And the cortex, which is the gray matter, where all the nerve cells are, lies on the outside. So it's like a walnut with fibers projecting inside. So initially it was thought that the white matter was the only target in MS. We now know that another very important target is the overlying cortex and the neurons, and we'll go over the details of that later. So really MS is a disease of the whole brain and of the whole spinal cord. But at least pathologically, it is most obvious in the white matter, but is not restricted to the white matter. At a microscopic level, what does this look like? So these are the nerve cells that we talked about, so that's where all the calculations are done. The dendrites are the connections that run into the nerve cells, and then each nerve cell has a single output through a fiber, an axon, and those collections of axons are called the white matter. So they are in the optic nerves, they are in the spinal cord, they are deep within the brain, and they are the vital connections that basically connect neurons with each other and with the rest of the body. If we go down even further, you can see that most of these fibers are ensheathed with myelin, and that's the key structure that gets damaged in multiple sclerosis. It's an insulator. Any wire that carries electricity, these are no different, has to be insulated. When that insulation is broken, the wire doesn't work. So it's really that simple. And why is wiring so important? So I like this analogy because most labs in the world, whether they work on stroke or MS or what have you, work on the nerve cells because they're the heroes of the brain. That's where the calculations are. They're the most sensitive. They're the most complex. But I make the analogy that if you have a telephone and its wire is disrupted and you can't plug it into the wall, what good is a fancy telephone if it can't communicate with the outside world? So that's why, from the beginning, in my lab, we focused on the wiring, the white matter of the central nervous system, and a variety of disorders, most recently in multiple sclerosis. And these wires, these axons in the brain and spinal cord are really quite amazing. If you think about it, they're serial devices, so they have to work perfectly from the beginning to the end of their connections. If you disrupt them at any point, they're out of the circuit. And I love this analogy because if you took an average 4-micron diameter fiber in a normal human adult spinal cord, maybe 1 meter long on average, and you expanded it to the width of a 4-lane highway, that axon would span from Halifax to Vancouver. And that axon works for 80 years, if there's no problem, of course, in our nervous system, which is really quite remarkable. And it has to work perfectly at every point. There is no road that I know of, at least in Canada, that works perfectly with no potholes or bridges out or anything for 80 years. So these are remarkable structures, and they're essential. So when you collect them together, that's the white matter, as I mentioned, and many diseases affect white matter. Here's some examples. Here's stroke, brain trauma. Here's little strokes that affect white matter. The little tiny strokes in the wrong place can give big problems. Of course, spinal cord injury is a huge problem because if you disrupt the white matter in the cord, like in the neck, for example, people become paraplegic. And of course, multiple sclerosis. Now, the difference between MS and all these others is MS is a very dynamic disease. So these other diseases happen once to unfortunate patients, but usually do not happen again, and then patients recover to a greater or lesser extent. But MS keeps coming, and it keeps coming. And the inflammation, the waves of inflammation as shown here, keep coming in the brain in a lot of patients in the relapsing remitting inflammatory form. Here's a particularly striking example of a patient who had an MRI scan done every week for a year. So every frame is 1 week, and you can see these inflammatory lesions coming and going week after week after week. Many of them are clinically silent, that is, the patient doesn't even know that they're happening. If they happen to land in a place in the brain or spinal cord or optic nerve that is very, as we call, eloquent, disrupting that, then patients get weakness or blindness or unsteadiness or what have you. So MS is a very dynamic disease. And the drugs we have are very good at controlling these recurrent waves of inflammation. And these waves of inflammation, of course, leave in their wake undoubtedly persistent damage to the underlying connections, and that's a big problem. So the approach was, well, if inflammation is a big problem, and if inflammation causes damage, let's control inflammation. And that's what current drugs do, and they do that very well. And if inflammation is not controlled, what happens with time, here is a recurrent scan of an MRI scan of the same patient over years. What happens is you get atrophy or a falling out of the brain volume. The same thing happens in the spinal cord. So early on in the disease, these are the ventricles, the fluid-filled spaces are small in young patients, and as time goes on with MS, the volume of the brain shrinks because of the disease, and in its place accumulates those fluid-filled spaces. So you can see the ventricles here are much larger here than over here, and what was lost was brain volume, mostly in the white matter, mostly in the connecting tracts. So inflammation undoubtedly accelerates this and promotes this. But the curious aspect of MS is that even with very powerful anti-inflammatory and immune suppressive drugs, this atrophy, by and large, continues with little change. So I think there's a message there, and we'll get to that a bit later. So these are the cardinal features of what goes on in MS. So here's a nerve axon, here's a cartoon of the myelin sheath that's surrounding it, and what happens in MS, if you look under the microscope, is that gradually the myelin is broken down for reasons that are unknown, and most importantly, the axons swell up and then they're cut. Now this is particularly bad because unlike in the peripheral nervous system, so if I injure a nerve in my hand, most of the times it'll grow back and I'll get recovery. If I injure a nerve in my spinal cord or in my brain, these nerves never recover. So once you lose these nerves in your brain or spinal cord, they're out of the circuit. And as I showed you from that cartoon of Canada, you disrupt that nerve at any point along its course and it is no longer able to function for you. So this is particularly bad when this happens. And it's bad because it's irreversible. When the myelin is lost, the brain has a certain capacity of replenishing and rewrapping these nerve fibers. So this is bad, but not as bad as losing the axon itself. And of course, early on in the disease at least, inflammation plays an important role. So when you look at these features under the microscope, and this has been done, if you look at the dates here, this has been done over 100 years ago, you see that. You see swollen nerve fibers, seen over here. You see disruptions of the myelin over here. The red should be continuous and it's disrupted. That's called demyelination. And of course, at least early on in the disease, in most patients, you see lots of inflammation. So all these dark purple cells are inflammatory cells that have come in and have caused an inflammatory response within the brain of these patients. So this is what MS looks like under the microscope. So for decades, we've watched this. We've looked with MRI, we've looked under the microscope, we've done biochemistry on blood and spinal fluids and whatnot, and what has kept coming back is inflammation, is autoimmunity, is inflammatory cells that are playing a huge picture. So it was quite natural, and I like this analogy, if you're sitting in the South Pacific on a battleship and you witness an atomic test on one of the islands in the South Pacific, and you from 20 kilometers away you see this huge mushroom cloud, it is quite natural to assume that the problem was in the huge mushroom cloud. And it's a little bit strange to dig in your heels and say, well, now wait a minute, even though this is an enormous mushroom cloud that I can see from 20 kilometers away, maybe something actually happened right down at ground zero. Maybe there's a little refrigerator-sized device that actually went off to cause this whole thing. So why would I focus here when the obvious thing is here? And this is what I think has been the issue with MS. It was so obviously an inflammatory autoimmune disease that that's where we have focused the majority of our investigations for the last several decades. And these investigations have yielded a tremendous amount of information about immunobiology, the biology of the immune system in humans, how it reacts in MS, what we can do to mitigate its anger and its attack on the brain and spinal cord, and therefore we've developed some very effective drugs to tone down and to quiet down that inappropriate angry immune attack against the brain and multiple sclerosis. But despite that, we still don't know the cause. You would think that if MS was a primarily immune disease, after all this effort, we'd have figured out at least the target of MS. We don't even know what molecular targets in the brain are affected in MS. We don't know what the cells and antibodies go after. We know they go after this and that, but there is no cause that we've identified. So I would then question, or at least wonder, whether a primary autoimmune cause of MS is valid. If after all this time and with very powerful drugs, and I'll give you some examples in a moment, we can't stop the disease, the atrophy continues, patients still progress in later phases. Even though we virtually reset the immune system, and some patients get bone marrow transplant, a complete reset of the immune system still does not cure the disease. I think there's a message there. So I would take this chicken and egg argument and say, well, maybe there's another model we should start thinking about. So the conventional model is what we call the outside-in model. There's a flaw in the peripheral immune system. That immune system attacks the brain, the myelin, the white matter, and you get MS. I would say, well, wait a minute, maybe there's something wrong inside the nervous system. And as a result, the peripheral immune system sees something wrong and then reacts to that. So I don't know who's right or who's wrong. I don't have any evidence for plan B. And that's what we're working on now. But I hope to have you walk away tonight at least wondering that I'm not necessarily wrong in what I'm saying. And the onus is on us to prove this, of course. So there's two competing theories now. The conventional one where it's immunity first and damage second. And the other theory that we like, that's a new one, it's different, unproven, I should emphasize. The inside-out model, where we're hypothesizing that there's something primarily wrong in the brain first. Just like in Alzheimer's disease, just like in Parkinson's disease, and that big mushroom cloud is a huge reaction to that problem. So if you kind of blow away the mushroom cloud with your anti-inflammatories, the underlying problem is still there. And that's what we're seeing with progressive MS. And I think there's a strong message there. So that's the fly in the ointment, is that drugs and very careful pathological examination don't really jive with a primary autoimmune cause for MS. And here's an example. So this is a pathological section from a human MS brain that was, for various reasons, obtained very early after an attack. And as you saw from the MRI, the cortex is on the outside, and below the cortex we have white matter. In this case it appears blue because it was stained with a stain, a Lux-Alphaz blue that loves the fat in the myelin sheaths, and when it sees that fat it turns blue. So all of this stuff should be dark, dark blue. And you can see this big blotch over here that has no blue at all, which means this lesion is completely demyelinated, and this is an acute MS plaque. Now, what's curious about this particular case is that when the pathologists looked very carefully in and around this plaque, they saw a lack of myelin, which is obvious even from here, but what they didn't see is a whole angry inflammatory reaction around this. So if you see such a profound loss of myelin and no inflammation, or little inflammation, and no immune cells, kind of makes you wonder if the immune cells are responsible for demyelination. And in certain cases they're absent. You kind of wonder, well, maybe the demyelination was really not caused by an immune assault on the brain. And it is cases like this that make me wonder and make me question the conventional assumption about what causes MS. Let's go deeper and more accurately with electron microscopy. So you'll remember this cartoon of the axon and its myelin. If we look under EM of a cross-section of a single axon, they're actually quite beautiful structures. So the axon is like this, and here you see this beautiful continuous wrap of myelin. The spacing is perfect, the continuity is perfect, the chemistry is perfect. I mean, these are really beautiful structures. And it is the integrity of this myelin sheath that is critical to allow nerve fibers to conduct quickly in an energy-efficient manner to make the whole nervous system work. So in MS patients, if you look very carefully at early lesions, you again start seeing some interesting anomalies. So here is an even greater magnified view. Here's the axon over here. Here's that myelin. You'll remember in the previous picture, that myelin was nice and compacted with a regular spacing of the little wraps of myelin. But look what we see here in this. This is a biopsy of an MS plaque from a patient. The problem, so this is normal here, nice and compacted. This spaced myelin is abnormal. It is decompacted for reasons we don't understand. But the problem here is, if you assume that immune cells have come at this nerve fiber from the outside and attacked it from the outside, wouldn't you expect the first problem to start on the outside of the fiber? Yet the first problem is starting on the inside of the fiber, where the immune cells don't even have access to. So that, when I see that, I start wondering, well, okay, that's interesting. That also doesn't jive with the outside-in model of MS. Not only that, when these investigators looked around this lesion, they didn't see any immune cells either. So here's an axon whose myelin is beginning to degenerate from the inside, and where there's no immune cell in sight at all. So when you see that, it makes me wonder what has caused this and whether it really is a primary external autoimmune attack. So another piece of the puzzle. When you start looking at treatment trials, these are even more interesting because these are real firm experiments in the human model of MS. These are patients that are treated with very powerful, very sophisticated, and very effective anti-inflammatory drugs that investigators and drug companies have developed. So these drugs are fantastic at halting inflammation and immune attack against the brain. In certain cases, in very advanced aggressive disease, as you know, patients have had their whole bone marrows transplanted. So these patients get chemotherapy, their bone marrows are basically destroyed, and then stem cells are re-injected to reconstitute a fresh new bone marrow, as if a newborn has had a fresh new bone marrow with no previous history of having seen any kind of antigens or things that might have stimulated that immune system to attack a brain in an MS patient. So these are very aggressive, very powerful treatments that work extremely well in shutting down inflammation. When you do MRIs on these patients, there's virtually no inflammation left in these brains. But there's a problem. So as a result, the relapses in these patients are also shut down. So people said, fantastic, we've solved the problem. But then if you look further and later, the problem continues. The problem continues in the sense of not inflammation or relapses. The problem continues with progression. So you've shut down one arm of the disease, the inflammatory relapsing arm. Excuse me. But the progression continues. And that was a big wake-up call for people and a big, important message for me. Because if we're so good at shutting down autoimmunity inflammation, why can't we fix the disease? Why can't we cure the disease? The progression, by and large, continues. So to me, the inescapable conclusion is in red over here. So you cannot argue that recurrent attacks of angry inflammation in the MS central nervous system is a bad thing, and likely leaves lasting damage. So if you shut down inflammation, that's always going to be a good thing. But because of all these features I just explained to you, pathologically, treatment, etc., these lingering progression and our inability to cure the disease despite all these aggressive efforts raises the distinct possibility that autoimmunity is not the cause, but perhaps a very inappropriate and a very important reaction to some underlying problem, the inside-out model that we're working on. So let me give you an example of why I might be right. So if you go through the literature and you look for case reports of patients that were diagnosed with MS, you come up with reports like this. So here's an example of a woman in her 40s, and for 10 years she's had a progressive problem with her gait. Speech became slurred. When her neurologist examined her, they found the slurred speech, they found her legs weak, they found her legs stiff and the reflexes off, and she had some sensory abnormalities in the lower legs. So visual evoked potentials, a test many of you are probably familiar with, confirmed that there's a problem in the visual conduction pathway, something we find very commonly in MS. Biochemistry and immunology tests from the spinal fluid confirmed that there's an immune response within the brain, within the CNS, something we find in 85% plus of patients with MS. And so this patient was diagnosed with primary progressive MS, a fairly typical example. She was treated with high-dose steroids and not unexpectedly had no improvement because this relatively non-inflammatory form of MS does not respond to steroids and anti-inflammatories. So she had no improvement. But I left out an important point. If you dig into the family history, this woman's son died at an early age with some severe congenital neurological problem of unknown cause. And this boy, on pathological examination, had a dysmyelinating leukodystrophy, which in plain English simply means a problem where the boy's myelin was degenerating in his brain. So that led these investigators to wonder, now wait a minute, this patient has MS and the boy who's genetically very close has something else? Probably not. Let's dig deeper. And when they did, they found that both mother and child carried a mutation in the PLP gene. This is a gene of the major protein in the myelin sheath. And the mother was heterozygous, which means she had one normal copy and one abnormal copy. So she presented later and had a milder form. The boy, unfortunately, was hemizygous, so this gene is on the X chromosome. So his Y chromosome did not carry a normal copy. So he was left with only the one mutated copy, which is why he had such a severe and unfortunately fatal form of this disease. But the point I'm trying to make is that here is a well-known, absolutely documented genetic, metabolic, and biochemical defect in myelin that in the mother presented with an absolutely typical presentation of progressive MS and was actually misdiagnosed as such. And there are many such cases in the literature. So the point of these slides is to show you that we can have known primary defects, something going wrong within the brain, within the myelin, within the white matter, that can, in certain patients, look very much like an MS presentation together with autoimmunity and inflammation and so on. So it makes you wonder if this disease that I just showed you was misdiagnosed as MS, could MS actually be some kind of degenerative disease where there's a, to date, unknown primary problem, and in fact the inflammation and autoimmunity that we commonly see is a secondary reaction to that. So if we're trying to unravel this, where do you start? I mean, this is a very complicated proposition, of course. The brain is a complicated thing. I learned that a long time ago, and I am humbled, really, by how complex neuroscience is and how complex our brains are. It's a fantastic field to be in. So let me spend a few minutes giving you just a very brief overview of some of the techniques that we use to study axons and myelin and try to unravel what goes wrong with them, at least in the laboratory. So I showed you this cartoon before. Here's an axon, here's a myelin sheath, some of the cells that support these fibers. So we developed a variety of sophisticated microscopic techniques to try to study and interrogate the very nitty-gritty of these fibers, the axon by itself, the myelin by itself, and so on. I'll give you a very brief overview. So first of all, the myelin. Myelin is tiny. It's only about 300 nanometers thick. That is half the wavelength of light that microscopes use to see things. So it is exceedingly small and very difficult to study, particularly in the living state. So we spent a number of years figuring out techniques to try to get microscopic signals out of living myelin so we can perturb that and see how it responds, see how it's made normally, and see how it degenerates and what we can do to mitigate that. So we use a variety of things, laser sources, we use ultra-fast pulse lasers. I don't have time to get into why, but they're very sophisticated. Here's one of our lasers, here's another laser. They're very neat devices, they're very expensive devices, but when you put it all together just right, they allow you to study and interrogate tissues, and especially axons and myelin, in ways that I never dreamt of when I was a student. So absolutely fascinating ways to marry engineering, photonics, and neuroscience, which we do in our lab. And when we did that, we were able to study myelin and axons in isolation. So I won't go into the details, and I'm not, actually this doesn't project too well, but trust me on this. So here's a living white matter tract under the microscope. The myelin is shown in blue, and we injected a dye that allows us to measure calcium levels in the myelin. Now, calcium is important in virtually every living cell because on the one hand, calcium allows normal biochemistry to proceed, but too much calcium kills, and it kills in stroke, it kills in MS, it kills in brain trauma. I'm not talking about the calcium in the blood, I'm talking about the tightly regulated calcium levels within cells. And no one's ever measured this within living myelin. We are the only ones that are able to do that. And when we did that, we found something very interesting. We found that myelin expresses transmitter receptors. Now, what are those? Those are proteins. They're pores that normally neurons use to communicate with each other. And nobody thought they existed in myelin. Why would they? Well, they do. And they're the exact same ones that are between nerve cells. And just like when nerve cells die in a stroke, myelin dies if you overactivate these receptors, and they're called NMD receptors. So it seems that the axon and myelin communicate with themselves through these receptors, and when they're overactivated, the myelin degenerates. So perhaps the demyelination that we see in MS may be due to some kind of aberrant overactivation of these very powerful receptors, and that's what we're working on now. The axon itself can respond to receptor activation. I won't go through all the details, but here are individual fibers from a spinal cord, and when we perturb them with different chemicals, we see increases in calcium shown here in the pseudocolor, and we were able to figure out that these axons, so not only the myelin, but the axon itself has receptors as well. So now we have an arrangement where, just like the stroke folks figured this out 15 years ago, that if you overactivate these, what are called the family of glutamate receptors, it kills neurons in stroke, we now show that if you overactivate these same receptors, you could A, kill axons, which is what happens in MS, and B, degenerate myelin, which is also what happens in MS. So now we have a new molecular framework in which to try to figure out why these two key structures degenerate, and most importantly, we have very powerful drugs to block overactivation of these receptors. So we're working on trying to tone them down and trying to reduce the degeneration of axons and myelin in various models, and hopefully in the real MS as well. So going back to the human histopathology, these swellings of axons have been known for a century, but we don't know why they happen. So we now show that the axons have these clusters shown here of these receptors, and we think when they're overactivated, they cause the nerve axons to swell, finally cut, which, as I told you at the beginning, is a very, very bad thing in the central nervous system. And this is a grad student of mine who's actually been able to model this human pathology. You can see here, this has been shown 100 years ago. We can now replicate this under the microscope in the dish, and showing how the nerve axon swells in green, the myelin here in red swells alone, sometimes one swells, sometimes the other. And we can control this and understand at a molecular level which of these receptors controls damage to each of these structures, with a view eventually to getting drugs to mitigate this. We use a variety of sophisticated techniques. We control polarization of light. You've all had polarized sunglasses, you know how that works. And you can see the myelin shown in red just by changing the polarization of light, of the laser light, which we control very accurately. You can change the configuration of how you see myelin. You might say, well, why would you want to do that? Well, that's because by knowing exactly what kind of laser light you're injecting and following the responses, we can infer the nanostructural, at the nanometric level, the organization of myelin. Remember that electron micrograph I showed you where the outside was compact and the inside myelin sheaths were separated and spread out because of something wrong in MS? Well, we can actually measure that with polarized laser light and infer things we can't see, but we can measure how the myelin is starting the very earliest points of myelin degeneration. Here's another fascinating technique. If you actually inject certain fluorescent chemical probes, we can interrogate the chemistry of myelin. So here's a collaboration with Wei Yang, with whom we work at the university, and they cause myelin damage in a spinal cord of a mouse, and we can measure the chemical changes in that myelin damage using what we call sylvatochromic probes and fluorescence spectroscopy. So I could speak for a whole hour on that, but you'd be all asleep. But anyway, it's fascinating stuff, extremely powerful, and we're able to study myelin and axons in very, very unique ways. The final technique I'm going to spend a couple of slides on because this is really neat, potentially very powerful. All of the previous techniques relied on our injecting or staining or infusing a fluorescent probe. So you have your tissue, you put a probe on, and then you see how it fluoresces, or the blue color we saw with that stain in that pathology section. But you can also get signals and beautiful images, as you'll see in a moment, without any probes whatsoever. And one of these techniques is called CARS. And here what you do, instead of one laser, if that wasn't hard enough, now we synchronize two laser beams at different colors. And when you do it right, chemical bonds start to resonate and emit photons of light for you, and you can make images from that. So this is a rather complicated technique. You can see this is one of our prototypes of our CARS microscope, so you need a physicist and engineers to work with you to get this all working, but as a result, you can generate absolutely spectacular images of nerve axons. So here's an example. There are no probes, no stains, no nothing. You just put it under this microscope, flip the switch, and you take images like this. You can see the exquisite detail this is the myelin in this case, and the axons are in between. This is a rat nerve. exquisite detail. And this is a human MS brain. And I show this one because this is our latest iteration of CARS microscopy called spectral CARS, where we can actually interrogate the chemistry of myelin changes in a human MS brain. An idea here is to try to understand the earliest chemical changes in lesions of MS brain using various techniques, in particular spectral CARS microscopy. And this probably doesn't project well, but the resolution of these images is absolutely spectacular. And by examining the real deal, human samples, we hope to get at the fundamental chemistry of what might be going wrong in MS brains even before any immune cell even smell that there's a problem in the brain, to try to prove our hypothesis that it's a primary defect in the myelin of the brain. So I'm not going to go over this, but just to show you that we and others have spent many, many years figuring out all the pathways that go wrong, that can go wrong, that will injure myelin and axons, and we're in a much better position now to suggest drugs to try to target certain aspects of these pathways to try to protect white matter in MS or stroke or spinal cord injury. So my last part of the talk is going to be very speculative. So this isn't necessarily true, it's speculation, it's hypothesis. We are working to prove it, we may be wrong. But so far nothing that I've seen out there in the literature suggests that there's no showstoppers for our position here. So I'm going to kind of ramble a bit and tell you what my view is of what I think the real multiple sclerosis is. So I showed you this image over here, and I explained to you why the scientific community has been so focused on the mushroom cloud, on the immune aspects of MS. And I've never done things the way other people do, which is why I'm looking exactly the opposite part of the mushroom cloud, which is ground zero, to try to figure out and prove or disprove whether there's a primary problem that actually caused the mushroom cloud, the inflammation, the autoimmunity to develop secondarily. So here's one possible model of what MS might be. So we've shown these transmitter receptors on axons and myelin. If you overactivate them, we know that myelin degenerates. When myelin degenerates, it spills fragments of proteins and lipids and whatnot because of activation of enzymes we know exist in myelin. We also know that myelin fragments are highly immunogenic, meaning when the immune system sees them, sees these fragments, it goes crazy and it reacts. We know that. And as a result, you have an inflammatory condition. And that inflammatory condition with inflammatory cells that rush in like that, they release a whole bunch of nasty things that in turn stimulate these receptors even more, and you get a vicious cycle. So the issue here is, so I don't know if this is true, but I think it's a reasonable model. So the point I want to make here is that the vast majority of MS research has been focused on this yellow oval here. We've been focusing on the immune response, the stimulation of autoimmunity, inflammation. We have not focused much on what might be going on outside this oval. And this is what we're working on. Of course, the big question is this question. If I'm right, well, you're quite reasonable. You'd be reasonable to ask me, well, what's the primary problem? What's the initial insult that triggers this whole thing? Something must have gone wrong at the very beginning to get this cycle going. Well, the short answer is I don't know, but we're working on that. But I think the point is that I think we have to think out of the oval, if you will, and maybe clues will come about this question mark down the road. But so far, that's a big unanswered question. And so I'll take this further. So one thing I can say is that everything we know about human MS, there is no single piece of evidence that will knock this down. Everything we know is still consistent with everything with this model. And therefore, I would propose that MS really starts over here, and that the inflammation that is so prominent is a reaction. And therefore, I'll take it further and say if that's the case, then the real MS is primary progressive MS. Because it just so happens that when you get MS, the vast majority of patients, as you know, end up with relapsing, remitting inflammatory disease. And that, to me, maybe, if I can be so bold, was a big distraction for the research community, that everybody started focusing on that. Everybody started focusing on the mushroom cloud which is perfectly natural. And we've learned a lot, and we've got a lot of good drugs for that mushroom cloud. But maybe, just maybe, the real disease is primary progressive disease, because in those patients, for whatever reason, there is no reaction, there is no mushroom cloud, and it looks like a degenerative disease, just like Alzheimer's and Parkinson's. And maybe we should be focusing on primary progressive disease, because it is simpler, and it hasn't been clouded and muddied by that very prominent and important inflammatory reaction. So my view of MS is this, and those with an engineering scientific background in the audience will know what convolution is. I view MS as a convolution, which is a smearing. So here's an example. You take two functions, the two white ones, if you convolve them and smear them, you get kind of a mishmash of the two in the red curve. So MS, to me, might be a convolution between some flaw in the immune system, because after all, patients with stroke or head trauma, they get myelin damage, but they don't get inflammatory MS-like diseases. So you have to have a problem with your immune system, coupled with, convolved with, some underlying degeneration that we don't fully understand. So this, to me, is MS. And depending on the balance between these two, you can explain all aspects of clinical MS. On the one hand, when the immunity, the flaw in the immune system is very weak, there is no flaw, you have primary progressive disease. Very little inflammation, progressive course, just like any other progressive neurodegenerative disease. On the other hand, as it turns out in most patients, you couple that with some flaw in the immune system, and now you have classical relapsing remitting disease. In the extreme case, you might know that the Marburg variant, these patients have a huge extreme inflammatory reaction. I mean, sometimes their brains look like they have a big brain tumor because the inflammatory reaction is so brisk. And that's called Marburg variant, also very rare. So that's at the other extreme, where the immune system is really angry at the first whiff of damaged myelin, goes crazy and causes a huge inflammatory reaction, looks like a big brain tumor. And as time goes on, and we know this also, through a process called immune senescence, the immune system kind of gets tired with age. And I think, I may be wrong, but I think that's why, with time, many, if not most patients, transition from relapsing remitting inflammatory disease to secondary progressive disease. Because the underlying degeneration, the blue line, continues, yet the immune system gets tired and stops reacting. So you go from relapsing remitting to progressive MS. So the important point here is that we're very good at stopping this. We know virtually nothing about this, have no drugs, no treatments, and the medical community has no options for this phase or this type of disease, and that's what we're working on. So we actually, this is the title of my talk, and we recently published a paper where it summarizes this kind of opinion of ours, this rambling. So if anyone's interested, please feel free to look it up. I can send you a PDF if you like. So this is where we explain this whole thing and why we think we are not necessarily wrong. So my last couple of slides, where does this leave us on practical terms? It's all great science, great speculation, et cetera, et cetera. Why is this important? Why should we be thinking this way? So through the arguments I just proposed to you, I think it's time to think of not only immune modulation, immune suppression, and inflammatory treatments, but it's time to think of what I initially called neuroprotection, but I now realize that MS is a pan-degenerative problem, that it's not only axons and nerve cells, it's myelin, it's the supporting glial cells. There are many, many cells that get damaged, MS in MS brain, and many cells degenerate with time. So cyto is Greek for cell, so I want to keep it generic. I don't know which cell yet. I have my suspicions which one is the primary target, but let's be generic and call it cellular protection, cytoprotection. So I think it's time to think about treatment, development of treatments to protect this cellular degeneration that is happening for some reason. And we are actively involved with international groups to try to push that forward. So really, anti-inflammatory therapy is here to stay. I mean, it works. And when I talk to my MS colleagues, patients do better on anti-inflammatory medications. So they're here to stay, and they work. Unlike other diseases, stroke, for example, where I spend most of my clinical time in, We have very favorable treatment time windows in MS. I mean, when stroke happens, it's minutes. If you don't have the drug in there within minutes, it's not going to work. And that was the big problem. Not so in MS. This is a chronic disease where you have days, weeks, months, years to get your treatments in to try to slow down the degeneration. Degeneration appears refractory to our best treatments right now, so this needs a lot of attention. And as I showed you from that cartoon, we are now much wiser than we were say 15, 20 years ago. We know a lot more about white matter, axons, and myelin at a cellular, molecular level, where we can come in and say, okay, you know, target this pathway, don't target that one because it's not going to work, and so on and so forth. So we know a lot more about the basics to try to propose and test new agents and new drugs in MS. So I'm going to stop here. I'm going to thank the many people who've worked in my lab over the years, worked very hard and generated these wonderful data, my collaborators, and of course the many granting agencies that have funded us generously over the years to generate these, to help us generate these interesting results. So thank you very much.
So I'd like to just say thanks to Dr. Stys. I think it's very exciting times in MS over the last couple of years for sure, and it certainly is heartening to myself as a person with MS that we've got a lot more investigation into the field that's going in different directions and trying different things. And to me, that's very, very exciting. So I'd just like to thank Dr. Stys for coming today, speaking about this, and giving us all a little bit of interest in a brand new direction. So thank you very much, Dr. Stys.
This concludes the Friday evening session. Lots of things for me to relay for you, to tell you for tomorrow. You have a choice. You can keep your name tag and take it home, but you've got to bring it back tomorrow. Or you're going to hand it in at the desk and pick it up tomorrow when you register. Also, on your way out the door, you can either go to the registration desk and pick up the red conference bag, or you can pick up the red conference bag tomorrow when you come back in. So it's up to you what you want to do. You need to have the tag, though, to get into the conference tomorrow, because these sessions are full. The dots on the name tag indicate which sessions